Okay, so we're live now. So hello everyone and welcome to the second session of the EANO Youngsters Journal Club. So I'm very excited. Before we jump into the paper, I'd just like to give a brief introduction on the format and on our goals. So these are my conflicts of interest. So the EANO Youngsters Journal Club is a 1-hour webinar discussing recent papers. So we want to cover the whole spectrum of neuro-oncology, from basic science to clinical trials, but also including all specialties from neuropathology to neurosurgery. We aim to provide a platform for lively discussions, but also networking opportunities between youngsters and experts in the field. So who is behind this journal club? So in fact, we founded a diverse working party of neuro-oncology aficionados where we formed the overall idea of having a journal club and also its implementation. We are always looking for motivated youngsters who want to contribute. So if you're interested, do not hesitate to drop an email or contact me via social media. Let me just briefly highlight some of EANO's activities, which might be interesting for you all as youngsters. So first of all, our annual meeting taking place in lovely Glasgow this year in October. Registrations are already open. Then the EANO Awards, where I'd like to specifically pinpoint the EANO Youngsters Award. So if you have published a great paper or know another one who did, do not hesitate to nominate someone. The deadline is at the end of June. You can find further information on that on the EANO website. Then just let me give some brief technical information. So you all as participants can pose questions via the Q&A feature. Just put in your questions, click or tap on send, and we will take the questions and discuss them. In the end, thank you all for your interest. Please share your feedback after the session. We will also send out a link to an evaluation questionnaire after the session. Now, let me introduce today's speakers. So first of all, Antonio Fuentes-Fayos, who did actually his PhD in Spain, but is now pursuing a postdoctoral fellowship at Cedars-Sinai in Los Angeles. He will present today's paper. Then Houtan Noushmehr, who is a PI at Henry Ford Hospital and Michigan State University in Detroit, who is the last author of the paper and will give us additional insights. Marjolein Geurts is a neuro-oncologist working in Rotterdam, so she will give some clinical insights and critical questions. Then as junior discussions, we have Mircea Tesileanu. He did his PhD in Rotterdam and is now a postdoc at Yale School of Medicine in New Haven in the US. And last but not least, Abigail Suwala, who is a fellow in neuropathology in Heidelberg. So with that, I'd like to hand over to Antonio.
I'm going to share my screen. Good morning, everyone. Thanks for the introduction, Max. Today, I'm going to dip into the epigenomic evolution of glioma using longitudinal data. So the article that I'm presenting today was recently published in Cancer Research, actually in the last March, titled "The Epigenetic Evolution of Glioma is Determined by the EDH1 Mutation Status and Treatment Regimen." The authors of this article take advantage of the GLASS consortium that have several number of samples well characterized across time, and they analyze 132 matched initial and recurrent tumor from patients with EDH wild type and EDH mutant glioma, integrating epigenomics, gene expression, and functional genomics. The results unveil that the standard treatments are related to loss of DNA methylation in EDH mutant glioma, resulting in epigenetic activation of genes associated with tumor progression and alteration in the microenvironment. Finally, they conclude that the treatment of EDH mutant glioma is associated with epigenomic evolution toward an EDH wild type-like phenotype. But before jumping to the result of the paper, I would like to introduce an important epigenetic concept in glioma, named GSM, glioma CpG island methylator phenotype. This phenotype is related with the chromatin remodeling and also with good clinical outcome. Also, it's associated with the presence of the 2-hydroxy glutarate that is the product of the EDH mutant enzyme. And because the importance of the epigenomics in the glioma progression, the authors in this article include and use the TCGA classification of the tumor based on DNA methylation, which divide the EDH mutant tumors between gold-deleted samples, GESIM high and GESIM low, and the EDH wild type in classic-like, mesenchymal-like, and LGM6 subtype. So starting with the results, the 132 samples were divided in EDH mutant and EDH wild type, and then they characterized the sample by DNA methylation, EDH mutation, TCGA subtypes and other subtypes, etc., comparing initial tumors with the recurrent sample. So zooming in this figure, we can observe that the EDH mutant samples have more loss of the methylation when they compare recurrent samples versus initial. And also in comparison with the EDH wild type, where this loss of the methylation was less than in the EDH mutant. Also, they didn't find too many subtype changes when they compare the initial TCGA subtype and the recurrent TCGA subtype. However, there is 20% of the sample that change in EDH mutant and 27% in EDH wild type. But even in the EDH wild type, the changes of the subtype is balanced. In the EDH mutant, exists a prevalence of the changes from GESIM high to GESIM low. Also, using another tumor classification, the pan-CNS based on DNA methylation, they found that the subtype changes become to be significant, since 40% of the EDH mutant samples switch to a high-grade astrocytoma. Also, remarkably, when they look the samples that switch subtype in the EDH mutant and also have hyper mutation data, they found that over 5 samples, 3 of them change to hypermutator status in the recurrent samples. And also 2 of these 3 samples change to high GESIM to low GESIM. That not happen when the subtype is retained, where over the 23 samples, only 1 change to hypermutator state. And also similar percentages are found in EDH wild type, despite if the subtype changes or not. So here I would like to do a break. If someone have any question to discuss, so briefly we can solve any question.
We haven't any question from the audience right now. Anyone from the panelist side? Yeah, Mircea, go ahead.
Go ahead. Okay.
Hi, thank you for the presentation. I think I have a more general question before we get into the nitty-gritty of the article, because I think on this panel, most of us are believers in DNA methylation, but when I talk to people outside, a lot of them would say, why is DNA methylation profiling really necessary? Isn't RNA sequencing more interesting because that's more downstream and maybe closer to the actual protein being made? So I was wondering, maybe Houtan, you can pitch in on this. If you could just explain for people outside of the DNA methylation field why this is actually an important topic to begin with.
Yeah, that's great. My camera's not working. I don't know why I'm not able to start it. It says host has stopped it. I was just wondering if the host can unstop my, yeah, okay. Okay, all right. So Mircea, that was a good question. I get that as well. But the thing about demethylation and epigenetics as a whole, because there's a variety of different things that can be called epigenetics, chromatin modifiers, transcription factors, anything that really modifies the DNA in some ways is called epigenetics. And if we think about epigenetics developmentally speaking, we can see that it's very cell specific and cell tissue specific, as opposed to, say, using RNA or DNA sequencing, because each tissue, although they have the exact same DNA sequence, the methylation is very different because it defines which genes are active, which genes are inactive. And so when we do classifications, we find that the epigenetics is a lot stronger in terms of its identification because of that cell specificity. And that gives us, and because it's almost a permanent mark, once it happens, it's very easy to pick up in terms of its sensitivity. So that gives us that idea or that ability to classify tumors. And when we looked at this in our 2010 paper, 2016 paper, and so forth, we found that the methylation was so much more stronger than any of the other molecular entities in terms of classification. So that gives us that information. And then in terms of the biology, there are other, when you look at mutations or RNA sequencing, methylation kind of defines which genes are active, which genes are inactive. And so when you see that in cancer, we see massive changes. But what we also do see is not only changes at the promoter genes, but we see them also in these intergenic regions, like these enhancer regions. And so I think maybe Antonio might go over this later on. This really can highlight other events that are very specific to the cancer progression. So epigenetics kind of gives us two flavors. It gives a classification. It can also gives us some biology context.
Maybe I can also say, as a neuropathologist who is from Heidelberg, where the Heidelberg classifier was also developed, which was also used in this paper, that what you just said, Houtan, it's great for classification. So it's the tool that we use, the #1 tool now, if we don't know how to, especially in brain tumors where you often have just limited material, so we can still use methylation and say what it is. But it's also great for giving sometimes a prognostic value, like in this paper, right? So for an IDH mutant gliomas, we see that they actually fall into two groups, the astrocytomas in low grade and high grade, when we say the Heidelberg classifier, or GSIM below, GSIM high. And so just for this classification purpose from the neuropathology side, it's just an amazing tool. And I think this paper is great because it just doesn't look just as classification, but beyond that, and to understand the biology behind that.
Yeah. Prognostic also, as well as predictive biomarkers that we can also find in some of these cases too. So prognosis is the top.
So thank you all very much for your input. As we don't have any other questions from the audience.
I think there's one question in the audience.
There was one question in the chat, but not in the Q&A. So there was the question whether there were any age-related differences. I don't know whether that was related to DNA methylation, but maybe Houtan, you can elaborate on that.
I'm not sure specifically which context this person is referring to, but I assume maybe the GSIM high, the GSIM low. We did not see any age differences between those two subclasses, if that's the question.
Great, thanks. So, Antonio, please proceed.
Cool. So all these data together presented suggest that the EDH mutant toward for a more aggressive phenotype and not the EDH wild type that looks like preserve more the initial phenotype. So in the next part of this article, the authors want to see the impact of this epigenomic evolution in the expression landscape. So they compare GESIM high and GESIM low samples using ChIP-seq sequencing to look for master regulators. And they observe that when they look into the transcription start site of the H3K27 acetylation and H3K4 methylation, they found several genes that have been related with cancer, like the genes, the HOX and FOX genes that, as I said, has been previously reported as oncogenes. Also, they identify the HOX T13 gene that is a transcription factor that have been associated with the maintenance and development of glioma. Also, when they look at more on detail on the A3K27N AC sequencing peaks and the A3K4 methylation, three match samples, in red the initial sample and in green the recurrent sample, they observe an increase in these peaks that reinforce the idea of the role of this gene in the tumor invasion, growth, and cell stemness. Also, they observe in the EDH mutant tumors with no correlation the overexpression of this gene that have been correlated with the stemness state of the samples. So they perform a CRISPR-Cas in vitro experiment using a EDH wild-type low GESIM cell line to knock out the expression of this gene. The host D13 knockout results in a decrease of the proliferation across time. So this data, we can conclude that the EDH mutant have loss of the methylation that could activate important genes such as host D13 and other oncogenes that could be related with the progression evolution of the glioma. So since the loss of the methylation in this sample were found after in the recurrence standpoint, the authors look for the influence of the treatment in this loss of the methylation. So they, also, I would like to point out that the treatment, the temozolomide and radiotherapy, have been reported as beneficial in the survival and clinical outcomes. However, the temozolomide have been associated with hypermutatus status and aneuploidy, and also the radiotherapy have been associated with deletions. So coming back to the results, the authors explore a subcohort of these samples that have been under treatment, around 36 samples with the temozolomide, radiotherapy, and the combined treatment, and also 33 samples that have been put in a watch and wait approach. Also, they use EDH wild type cohort. So the authors identify 620 CPGs that's correlated with the loss of the methylation in the EDH mutant samples that have been put under treatment. And they found that in the recurrent sample, they lost the methylation when they compare with the initial. Also, the non-treated patient showed the same methylation pattern in these 620 CPG sites, and also in the EDH wild type the treatment doesn't make the loss of the methylation, and the pattern of this methylation looks like in the EDH mutant treated patients. Also, this data was validated in an external cohort, also from the GLASS consortium in Netherlands, and they found the same evidence that the EDH mutant samples lost the methylation after treatment. Also, by comparing clinical and molecular data, they discovered that a significant percentage of the samples progressed to high-grade tumors after treatment. Also, in these samples, a considerable percentage progressed to GSM high to GSM low, and also they changed the pan-CNS classification, the Heidelberg classification, and the hypermutator status. So to see the biological influence of this loss of the methylation, they compare and correlate DNA methylation data, DNA sequence motif data, and gene expression. So the authors identified 24 CPGs associated with the neuro-D1 motif, resulting in 18 genes potentially regulated by DNA methylation after treatment. Among them, we can find known oncogenic genes like MIP that have been associated with the control of the cell survival and progression and proliferation, and RSPO4 that is an agonist of the Wnt beta-catenin pathway. So continue with the article results. They take advantage of this DNA methylation data, and they use these data to deconvolute cell subtypes in the tumor microenvironment. And they found, when they compare treated and non-treated samples, that exists an increase in the treated sample at the recurrent point of the endothelial cells cluster and the CD8 positive T cells. Also, this was validated by immunohistochemistry. So here we see that the treatment could have potential to modulate the tumor microenvironment and the angiogenesis. And finally, the author looked at the survival data with different strategies. First, they study that the patient with that have been treated after the first surgery have worse survival than those that have not been treated. Even the mean are close, the treated patient have poor survival, or have worse survival. Also, when they look in the progression-free interval between the first surgery and the second surgery, the treated patients look to progress more, so have better survival than those that have not been treated. And finally, in the last analysis, they found that the progression-free survival from the second surgery to the last follow-up, so death or progression, the treated patient again show worse survival. So to the end, I think that we can finish and then discuss. As a study conclusion, exists a loss of DNA methylation associated with the standard treatment and EDH mutant tumor, which cause epigenetic activation of genes associated with early tumor progression, such as HOXD13, and alteration in the tumor microenvironment context toward angiogenesis and T cell composition. Also, the molecular profile of the treated EDH mutant tumors resemble treatment-naive EDH wild-type tumors. And finally, in treated EDH mutant tumor progress later than the treated ones, either spontaneously or after subsequent treatment. So thank you to join to this journal club today, and let's follow with the discussion.
Great. Thanks a lot, Antonio, for this great presentation.
Welcome.
I already have one question from the audience, which is a bit complex. Maybe I might forward it directly to Houtan as he was involved. As Hox family members are glioma stem cell related genes with their promoters hypomethylated in glioma stem cells. How do methylation profiles of glioma stem cell reservoirs evolve by recurrence in IDH mutant and IDH wild type cases? Do you have any data on that?
So the question is about the tumor cells or stem cells?
Yeah.
Specifically, we didn't look at that. We explored the association of stem cells using markers for stem cell by DNA methylation, and we found that there was an association with stem-like features in these GSIMP low phenotypes, but we didn't specifically look at stem cells in the brain, if that was the question.
Thank you very much.
Yeah.
Currently, I don't have questions from the audience, so please put in your questions there. But Marjolein has a comment or a question.
Yes, thank you. So I'm a clinician, and I must say that I'm having a hard time interpreting these data from a clinical perspective.
Oh, that's not good.
It won't surprise you. So let's have a look at the subgroup of the IDH mutant tumors in this paper. You're describing 69 patients who've had treatment. 33 of them did not receive treatment, so they had a wait and scan policy, and 36 of them received radiotherapy or temozolomide or a combination of both.
Right.
And now we're finding and we're seeing data that the patients that were treated have a decrease in their DNA methylation levels.
Right.
Now, I'm wondering whether we're actually looking at an effect of the treatment or that we're treating bad tumors.
I'm sorry, repeat that again. What was the question?
I will. The actual question is the treatment bad, or is the tumor that is treated bad?
Oh, well, that's a good question. I guess you're asking whether there's a predisposition to this type of phenomenon, right? I guess I can't say for sure what that might be, but it does seem like any type of treatment, whether there's chemo or radiotherapy or combination, all we are seeing is a loss of demethylation. We're seeing this phenotype that looks very similar to the IDH wild type, which is kind of like they're more highly aggressive, high-grade gliomas. And so now there are questions, right? So maybe one of the good questions could be asked is, are there subclones that are maybe resistant to therapy that are emerging in this data set? So that's one idea that might be emerging, that there might be a mixture of IDH mutants and IDH wild types in the tumor cells at the GSIMP high level at the initial diagnosis. And when you treat them, those that are more prone to treated, they would get removed, and those that are resistant, maybe those are the IDH wild type clones that emerge, and that then projects this hypomethylator phenotype.
Yeah.
So I don't know. I suspect there might be subclones that are resistant to therapy that are emerging in this data set.
Yeah, I see your explanation, and I'm just trying to transfer that to clinical practice. So I'm in this multidisciplinary board, and I have to decide whether or not I should treat this patient? Well, I think based on your data, we're not doing quite a bad job at this moment. Based on rapid tumor growth, MRI findings, gut feeling, maybe even, we decide which patients we treat and which we won't. And now we find that the patients that were treated, at least in the class cohort, were indeed performing biologically worse. So could this association that you're showing maybe be turned around and used in the opposite way as a predictive marker, as you were already suggesting, for deciding which patients would need treatment?
Yeah, that's a good question. Like in 2018, we published a paper where we came up with predicted biomarkers that can predict those patients that would become GSIMP low. At that time, we didn't have treatment data, so we couldn't associate that information with what we were finding. But in that paper, and Mircea knows it very well, we validated in the Catenin project, where we looked at predicted biomarkers that can predict these type of patients that would then emerge into the GSIMP low. So yeah, so that's another advantage of using demethyllation, is that you can get these ideas of not only prognosis but prediction as well.
So in short, do you agree that we still need to give our patients treatment if we, well, as of now, like tomorrow in our tumor board, can I just go on and treat them?
That's a very difficult question, and I reserve, I have reservations. I'm not a clinician. I'm just a scientist, so I can't tell you for sure what to do as a clinician. I think there's need to be more data out there to really confirm this, and I think maybe a proper clinical trial is probably needed, to maybe assess this. But I can't tell you, what's the best situation here in this case. All I can show you is that there's a strong association with treatment and response, both molecularly and clinically. Yeah, I don't know how to tell you how to answer that question specifically, but
well, until then, maybe we should just rely on the great clinical trials that are already out there.
Yeah.
Yeah.
I mean, there's only chemo, radiotherapy, and surgery. I mean, there's nothing much more you guys can do. And I as a clinician, I mean, not if I were a clinician talking to a patient to tell them, man, there's really nothing else to do. I don't know if the patients would like that, right? I don't know if they want to hear no treatment. I don't know. As a patient, I probably wouldn't want to hear that.
Thank you.
Maybe if I can add something to this point. So I think this is a very good point, Marjolein, and I think that was also mentioned in the discussion of the paper that maybe the tumors that initially didn't get treatment or had the watch and wait treatment were initially looked like histologically better than the ones that got treatment. And I looked a little bit specifically more into one of the figures where the WHO grades were listed, and it seemed like really the tumors that were not treated or got the watch and wait treatment were more WHO grade 2 tumors, right? So this is then also related to that. But I think this paper also raises another interesting point, and that's, Houtan, what you also mentioned is that we see in the tumors, the IDH mutant tumors that progress, that they become more like IDH wild type tumors. They resemble them at the epigenetic and maybe also transcriptomic level. So the question here is, maybe the tumors that progress, what has happened? Maybe the IDH mutation is just losing its importance in these tumors. And maybe that's why they lose this hypermethylated phenotype. Yeah. And I'm raising this question and maybe this is an idea, and I would like to know what you think. Now that we start to treat patients with IDH inhibitors, especially low-grade patients, should we also really use them for high-grade tumors?
You mean IDH mutant high grades?
Yes, of course.
Yeah.
Yeah, course. I mean, like WHO grade 3, 4 tumors, astrocytomas that are IDH mutant.
Yeah, wow, that's a good question. I don't know how to answer that question as well. That's a very provocative question, but yeah, I think inhibitors, the IDH mutant inhibitors, show some promise, you know, in the clinical trials. So, you know, I think that there's some opportunities there, but yeah, I don't know how else to answer that question. Sorry. Antonio, did you have a question?
Yeah, Antonio.
Yeah, I have a question for Houtan. Like as a translational scientist, I want to know your opinion about why when we saw the tumor here, the EDH mutant tumor, they progressed to like a really aggressive phenotype, but then that is not reflected in the survival. So what are your thoughts that is happening between the bench and the clinical practice?
Mm-hmm.
Like it's related with the Marjolein question, but
Mm-hmm.
because this,
Mm-hmm.
I think that this paper is really good opening a lot of future projects, but
Do you
I want to
see
know
a
your
lot of
opinion about that.
clinical trial opportunities here?
Yeah, and also like basic science, like as also I love wet lab.
Okay.
And when I saw the experiment of the CRISPR-Cas, I have a lot of questions about that, like which Cas you use, why this cell line that you really explained really well in the paper, anyway,
Yeah.
about the limitation, but why not other model like organoids or whatever?
Yeah.
So
So that's another good question as well. You know, we didn't have an opportunity to. So when we published the paper, reviewers were asking for validation experiments, so we had to do something kind of quickly. We didn't have time to do
yeah,
organoids and other things like that. You know, so obviously, like as we put in the paper, you know, further studies are needed. We would love to have used other cell line models, but as many of you know, IDH mutants are very difficult to grow in culture. And so we were very limited to what we could use. And that was a big challenge for us as well. Again, I think there's a lot of opportunities here to study this. And you know, and to get back to why epigenetics is so important, you know, and why look at that versus look at RNA or DNA. You know, with epigenetics, you can homogenize the samples. You know, with RNA, they're very heterogeneous. You know, it's hard to dissociate and separate groups of patients into prognostic classifiers. But with methylation, you can put these patients into a more homogeneous population, and then you can do very focused studies. You know, and to do like these experiments that you want to do, so you're not getting a lot of noise or confounders. So that's another advantage of the demethylation era of this data, or looking at it in terms of unique cancer types, in particular gliomas. Gliomas are very specific. With epigenetics, it seems like it's just because of the IDH1 mutation that kind of creates this phenotype. It's, you know, when we look at this across 33 different tumor types through TCGA, glioma has the highest overall methylation level. And so epigenetics seems to be a very important factor in this tumor type, and so it's worth studying, I think. And so, you know, we did chip sequencing. We looked at the EREGs, which are these epigenetically regulated gene sets. There's lots of opportunities to look at the biology of this using DNA methylation. So it gives us two flavors. It gives us an opportunity to classify tumors, and then also gives us the opportunity to look at the biology. And you see from this paper that the methylation data can really put patients into different, very homogeneous buckets that refines the prognosis, you know, in a different aspect. I
thank
don't
you.
know if that answered your question, Antonio, but
Thank you very
Yeah,
much.
thank you.
We have one question from the audience. Maisam.
Can I first maybe add something on this last question?
Yeah.
Because I also thought it was an interesting question why you may have progression to something biologically worse, which clinically might not be shown in the survival. Could maybe also a part of this be the inherent selection bias of glass? In the end, it's a retrospective cohort for which a patient needs to receive 2 independent surgeries. Could it be that maybe the patients that went on to have a worse biological phenotype usually do not make it to the second surgery and are therefore not seen in this cohort?
I think it's the other way around, Mircea. People with a good prognosis may make it to the second surgery, and I think this is reflected in the fact that there's less oligos than expected in this cohort.
Yeah, so that's kind of what I mean, is that the ones that have like a bad phenotype biologically here, but are prognostically better, are maybe different from the actual bad ones, which have already passed away. Maybe that's the reason why we don't see the survival differences.
Could be. Yeah, there's quite some selection in this cohort, as always in this retrospective series. So I think you need to be super careful with interpretations when it comes to survival, etc. But the biological insights are invaluable, I think.
Yeah. I agree.
Yeah, there were questions about resection, the extent of resection, grading. You know, these kind of things can also be a factor in terms of their outcome. So that does. We try to explore as many covariates as we can by looking at, you know, grading and age, as well as extent of resection and things like that. So again, you know, I think a proper clinical trial is needed, really, to really make this a clear point, you know, to make sure that this is actually accurate or not. But yeah, I think there's some opportunities here for follow-ups.
And how would this clinical trial look like, in your opinion?
That's a good question. I don't know.
What would the aim be? What question do we need to answer?
Is treatment necessary?
Yeah. So I may be a little biased Rotterdam-wise, but I think we have the Houtan trial showing that adding temozolomide in the IDH mutant tumors to radiotherapy gives the answer. Mircea, Mr. Houtan, what do you think?
And yes, I think I agree on that part. What maybe could be interesting is prospectively taking samples. I mean, surgeries would be a bit much, but maybe liquid biopsies to see how this progression goes, independent of any treatment. I think that could be a good step.
Yeah. What do you mean by liquid biopsy?
Liquid biopsies, for example, by a lumbar puncture, to see if we can find this methylation differences in these samples, and then if you already
prior to surgery or something.
Yeah, so prior to surgery, and then let's say a year later, so you have a specific time set to actually compare it to. Which all the ones included would be falling into.
There is then one question from the audience. So Maisam Motevaseli asks, would increased infiltration of CD8 positive T cells in recurrent cases be related to the subtype switch to the mesenchymal phenotype, which is more largely dependent on the tumor microenvironment?
That's a good question. Yeah. Well, we didn't see any subtype to this IDH wild type mesenchymal-like phenotype, but that's potential. That's, you know, it probably goes back to this idea that they are very IDH wild type like phenotype, you know. So Yeah, good question.
Then there is another question. Thanks for the excellent presentation. Pablo Vallecosa writes, is there any impact on the efficacy of drugs such as vorasidenib as a treatment for IDH mutants? So maybe based on the methylation profile, is there any data on that you are aware of?
Not that I know of, no. Clinicians on the call would know, I don't know.
I think it also refers to Abigail's question a bit earlier, like what do we know of IDH inhibitors in which tumors, in which patients will benefit? I think there's very little we know until now.
Are you guys doing IDH inhibitors to your patients?
Yeah, in the Netherlands we have this patient access program, so we've treated a few patients.
Oh, and how did they respond?
Well, it's only 6 weeks since it opened.
Oh, okay.
So I think we're still stuck with the Indigo results, which are promising, I think, as we all know. But what is nice, I think, what we can learn from the studies that preceded Indigo, like the phase 1 studies with the Evo and vorasidenib, is that it's not only the grade 2s non-enhancing responding. There were also contrast enhancing and higher grade and even recurrent tumors responding, but just not as pronounced as the non-enhancing grade 2s. So my interpretation of that is that these like early phase IDH mutant tumors, very sorry for this bold statement, are probably responding better than later stage, but we have to find out, I think. What I wonder is whether, and that relates to this, is whether the patients that have been treated with IDH inhibitors and ultimately progress under IDH inhibition will still respond to temozolomide. And I'm not sure whether there's people in this panel who have an opinion on that.
Why do you think they would not?
Well, if we now see in this paper that there's under progression patients who lose their IDH mutation, and I think we've seen that before in other sets, then maybe selection for the IDH mutation via IDH inhibitors may make that happen even more. So we end up with IDH wild type tumors being less responsive to temozolomide.
But also there are patients that haven't methylated MGMT promoter do respond. Mm-hmm. So yeah, I think this is a very interesting question. But I think at that point we can only just,
speculate.
Speculate, exactly. But which is obviously not very, doesn't make us very happy. But I think, it is a very interesting question, this one, and also what is happening with the patients that we treat now with IDH inhibitors. Do they gain this CGIMP high phenotype, CGIMP low phenotype, so high grade and being hypomethylated? Do they gain this phenotype more quickly because they don't have the IDH mutation, and do they become more aggressive more quickly? I think this is very interesting because, please tell me if I'm wrong, but the Indigo trial, it was not overall survival, but for progression-free survival, right? That was taken into account. And I think I looked at the earlier trials or studies before, and they saw that, 2-hydroxyglutarate goes down when they treat the patient. So it's exactly what we would expect for a higher grade astrocytoma.
There is then another question coming from Angelo Di Pasquale. He writes, "Thank you for the presentation. Probably randomized trials about overall survival advantage of radiotherapy chemotherapy in IDH mutant gliomas are the best evidence we have to date for clinical practice. Maybe the methylation profile could contribute in assessing the risk profile of our patients at diagnosis and in choosing the best strategy, wait and see versus vorasidenib versus radiotherapy chemotherapy." So, yeah, actually, I think a very good comment that just underscores that we just need more data. So we need clinical trials with a strong translational research foundation to be able to answer those questions. Okay.
So maybe I can just say from like the neuropathology in Heidelberg, we obviously already do DNA methylation arrays for all the patients, and with IDH mutant gliomas also to just check for the 1p19q status. So we do it and we take this into account. So when we have a tumor that looks based on histology, an astrocytoma like WHO grade 2, but in the methylation profile, we see that it falls more into this high-grade group. This is something we take into consideration when we do our final diagnosis.
That's very nice. It's nice to know that methylation has come a long way.
Then I have another quick question. In the results section of the paper, I noticed that there were some cases who switched the subtype, so from astrocytoma to oligodendroglioma or vice versa. And there were even 2 cases who lost the IDH mutation. Is there any explanation on that? Houtan, maybe?
Yeah, that's a good question. I mean, Joe Costello showed that a while back ago, that there are some tumors that lose their methylation in the recurrence. I don't know what could explain that. I would think maybe there are subclones that are resistant to therapy that might, you know, evade it and then emerge, and then those are the ones that are these IDH wild type subtypes. You know? I don't know.
Because actually, correct me if I'm wrong, but the IDH mutation and also 1p19q co-deletion are very early events in genesis. So actually, it's a bit counterintuitive that the tumor is then able to lose those factors.
Yeah, that's a good point. Yeah, I don't know where we would stand with that. Yeah, that's a good question.
Maybe then, while we are waiting for other questions from the audience, I have a question to Abigail as neuropathologist. Is there any role of methylation in IDH wild-type glioblastoma to stratify prognosis or support us clinicians for decisions on clinical practice?
That's a good question. So right now for IDH wild-type glioblastomas, we don't see a difference in survival when we look at the different methylation groups. So now speaking of the Heidelberg classifier, I know there are other classifiers, but we mainly put them into 2 groups, mesenchymal RTK1 and RTK2. So from these 3 groups, we don't see that there's a difference in survival. What we notice is that more recurrent glioblastomas fall into the mesenchymal subgroup. So this is definitely something we can see. We usually, when we do DNA methylation on these tumors, it's because we, for example, want to just be sure that it's not any other tumor. Yeah, because with IDH mutant gliomas, we have certain tumors that it can be, but with IDH wild type, it's sometimes there are different entities that have also different prognosis. So this is why we still often do DNA methylation, especially, for example, in younger patients, or if there's something or that is somehow striking about the patient's clinical history, just to be sure that it's really glioblastoma. But from the prognostic or also predictive point of view, there is not really a reason why we use DNA methylation, except for, of course, that we can also look at the MGMT promoter, because it's a specific site of methylation that we can also see. And of course, we can look for copy number variations, like also the CDKN2AB status, or also amplifications like EGFR or others that could be targets for molecular therapies.
Marjolein, do you have a comment on that?
I have a question, I think, to Abigail. When will we find methylation in the WHO classification?
Well, we already do. So with 2021, there are certain entities that you can basically just classify with DNA methylation, like the H gaps, for example. And also for other entities where DNA methylation, if you use the classifier or also the classifier from Bethesda, and you can, with a higher insurance, say it's this entity based on DNA methylation, you can use that to do your integrated diagnosis. So I think DNA methylation already made its point into the WHO classification, and I think that yeah, it's will probably only be maybe proceed that. Maybe we also have new clinical assays, molecular assays, which I find very exciting, that will come up with the next WHO classification. Of course, it's always, we always have to take in mind that not every site is able to perform all of these, right? So this is a drawback. But I think whenever we come up with a new methodology, new assay that really shows us it's really better for classification than what we have so far, then at some point it will also make it into WHO classification.
And just to add to that, you said that maybe the DNA methylation profiling cannot be performed everywhere, but one of the advantages also of this assay as compared to the RNA-seq is that DNA is much more stable, much easier to send to a specialized lab, because we don't need every center to be able to do everything if we could collaborate a bit more.
Okay, I think there is time for one last question or comment. Maybe also from panelist side.
I think the last sentence with "we should all collaborate more" just was a very nice last phrase.
Agree.
Yeah. Agree also with that, and also agree again that we just need more data, more clinical trials with a strong foundation of translational research to tackle those difficult questions. So yeah, thank you all for participating. Thanks also you as panelists and you, Antonio, for presenting this great paper. So it was great to have you all here. It was a great hour of discussion of recent data. So please, all who participated, please fill in our evaluation survey. I just put in the link for the evaluation survey in the chat, but you'll also get a link for that in your participation confirmation email that you get from the EANO office. So with that, thank you all again, and happy to see you at our next Journal Club session. Have a nice evening, everyone.
Bye. Thank you.
Bye.
Bye.
Thank you, bye.